Yes. So, ideally, this is what we had to discuss. That is, first, like the chronological order that I will go with is first the discussion session and then the PYQs. Because last week also, if you remember, most of the PYQs, like the logic behind what the answer was and the rationale behind it, had already been discussed in the live session. But there were a few that we couldn't, get through last time also. So that we'll pick up right now. But once we go through these discussions prior to the questions, you'll have a better understanding of it. And even if right now you are not that prepared to answer those questions, I hope by the end of the discussion part, at least you will have a better understanding of that also. Right. So, for instance, we'll be discussing about some ions, for example, right? Just a moment. Yeah. So, for instance, we'll be discussing about some kind of an ion, right? So, over here, what or how ion channels are, what is a refractory phase, what is a ligand by, bound ion channel, what are the other kinds of ion channels that are there, right? So, if we have already gone through that during the discussion, it will be easier for you to answer those questions also, right? And it helps you, sort of build this concept also, that once you read the thing and then you solve questions while that thing is fresh in your head, it will help you retain these concepts also. So that is what we are aiming to do. Yeah, but before this, as I said, there was some part. Okay, I'm unable to escape. Yeah. So there was some part from your previous year also, from your previous lecture also, that was left, right? So this is actually the last slide that we had done. We had begun with explaining the different kinds of ion channels and how there are different kinds of conformational changes that they sometimes go through, or they differ in structure altogether, corresponding to different kinds of ligands that are there, or different kind of mechanical pressure, or some other, cascade of ions altogether that uh, help them be in either an open state or a closed state, right? So in that regard, we had gone through these three kinds of ion channels. One was a kind of ion channel which has a conformational change in one region. So which is the region over here? It is this region, right? So it is like a gate. So once you come inside, the gate closes, right? So once whatever was the allocated amount of ions that had to go in or come out of this channel, once that is attained, this gate, it closes, right? Until and unless another burst or another bout of these ions are eligible to go out or come in, right? Then there could be a general structural change also, right? So instead of the gate closing, you can think of it as at night you are using your top sheet, your blanket on top of you, and in the morning you just fold it and you just keep it on the side, right? So the entire structure of that blanket has changed now. It is no longer spread, no longer open. It is now closed and wrapped up, right? So that kind of changes also occur in these ion channels. There are yet other kinds of ion channels which could have a blocking material or something that outside of the ion channel itself has a conformational effect on it, right? So over here the channel remains the same, right? But there is some other protein that is attached to its tail that is coming and blocking the passage, right? So because of this, again, the flow of materials and ions might be halted, right? Unless and until they have a very solid reason to go out, or they have a good enough flux or a gradient built already, right? Then this slide also we have done that there are several types of stimuli that control the opening and closing of ion channels, right? These could be ligand gated. What does ligand means? It basically means a particle or a chemical substance that comes and binds to the active site of a receptor, or it binds to a receptor, and and the response is that this channel opens or it changes in conformation such that opening of the channel occurs, right? It could also be voltage gated. Now, how will this occur? For instance, this channel was open, right? And because of it being open, a lot of sodium actually went inside, potassium went outside, chloride also went outside, right? What happened as a result of this? The potential difference got shifted, right? Initially, outside it was negative and inside it was positive. That is not initially, actually, inside it is negative and outside it is positive. So let us take this as the normal condition, right? So initially, this was the case, but due to some movement, maybe through some other channel, now this is the case, right? That outside it has become more negative and inside in relation to that or in regard to that it has become more positive, right? So now there is some instability that is there. It needs to bring it to an equilibrium state. That is why uh, based on this voltage change, this channel might open and allow passage of ions back to their original places, right? And then there could be stretch or pressure gated channels. Now, these are the kind of channels that are, for instance, present in our skin, near our palms, in our fingers, etc. And they respond to mechanical pressure, right? So, mechanical pressure basically makes them open. Under the influence of these regulators, basically, what are the regulators over here? They could be the ligands or the mechanical pressure or the voltage difference, right? The channels enter one of the following states, right? So if the channel is closed, but it can be activated, right? That is known as the resting state, right? So this slide is important for us to then understand how action potential is actually attained, right? So pay a lot of attention to this slide for that, right? So if it is closed, but we can activate it if we give the ligand or if we apply more pressure, it can be activated. Then that is known as the resting state, right? If on the contrary, uh, the channel is open, right? Then it is already active. There is a third possibility wherein the channel could be closed, but in this state, it is non-activable, right? So whatever amount of pressure you apply, whatever amount of, voltage gets changed or ligand you give, it will not open, right? So it is closed and we cannot open it, right? So that is known as the refractory phase, right? So, the techniques of how we have actually been able to look at these various phases and look at action potential actually is given over here. It is through the patch recordings, wherein single ion channel cells, ion channels from the plasma membrane of the neurons are actually recorded, right? This is kind of what happens. So this is a cell view. This is the cell body of the neuron in the culture dish. And over here, you can see this triangular light that is coming, right? So this actually is a needle. It is a very, very fine needle, but it is at 200 to 600x magnification. So that is why it looks like this, right? Plus the, angle is not like its plane is not the same as the cell body of the neuron. That is why it looks as if it is not focused over here. So, but what is happening actually is depicted in this, schematic. That is that, for instance, an acetylcholine receptor or one of the ion channel receptors is, picked up by the end of this very, very thin needle. Right. And from that, there is a measurement that is taken, which is the measurement of the ion flow through that channel. Right. So this is known as a patch recording. Yeah. And this could be the output of such a recording, right? So if this line depicts the channel being closed, this line depicts the channel being open, right? So over here, you can see that there was a significant amount of time when the channel was open, and in between that, there was a burst in which it was open also, right? like these are the different kinds of recordings that you can get from the channel, right? This is an example from an acetylcholine gated channel. Now, when we look at these recordings, right, we can see that the opening and closing of a single channel is binary and probabilistic event, right? So it depends on triggers. So why are we saying it is binary and probabilistic? Because it is either on this line or on that line, right? So if you see in the previous slide, this is closed, this is open. There will be no partly open or partly closed over here, right? So peaks, why we are looking at these peaks is a little different thing, right? That is maybe the frequency with that it is continuing to be open or it is continuing to be closed, but it is not lying somewhere half closed or half open, right? So it is binary in that regard, right? Now, why are we looking at these individual peaks? Because they sometimes we can take an average of multiple channels actually, even in these patch recordings, right? So average of multiple channels can show smooth transitions and larger tip size like this, right? But if it is a single channel, you will always see either that line or this line, right? This kind of transition will be missing. If this kind of transition is present, then you can easily, it is safe to say that multiple channels are being recorded over here. It is not a single channel recording, right? So, yeah. This is a cell attached kind of whole cell patch configuration, and there, sorry, this is a cell attached kind of patch clamp, and this is a whole cell kind of patch clamp. Now, what is a major difference that you can see over here? Initially, the cell attached kind of configuration is what is always achieved when that micro pipette or micro tip or micro needle is actually placed on a single channel and that recording is taken, right? Sometimes what also happens is that whatever this tip is, right, we know the kind of channels we are targeting. If we are targeting acetylcholine channels, then synthetic acetylcholine we will fill in this tip, right? So that if and then we will apply a suction pressure so that the like this membrane, it kinds of merges with the pipette, right? So, and the insides of the cell body, they mix with the contents of the pipette, right? And then if the recording is taken, it is for the nearby channels also, right? And that is the whole cell recording basically. Yes. So there are certain properties of an action potential that we want to study over here, right? And to understand this only, whatever has been told to you regarding the ion channels, regarding the, resting and refractory phase, regarding these recordings, different kinds of recordings has been told to you, right? So this is an important phenomena to study of how an action potential is attained and why does it behave like this, right? A strong, sharp peak. And it has some other properties also, right? So what are the properties of action potentials? There is a threshold for action potential to occur, right? So if a threshold is crossed, only then action potential can be achieved. If that threshold is not crossed, then this strong sharp peak will not come, and only this kind of plateaued curves will come, which will be very, very tiny, and they are known as electrotonic potentials and not action potentials, right? It is an all or none phenomena. That is, either it will occur or it will not occur, right? That is why it is binary also, right? Either it will be on this end or this end, right? It will not be somewhere in the middle, right? Half an action potential will not occur, right? That it is partly undergoing action potential. You will never hear that. Either it is undergoing action potential or it is not, right? So it is an all or none phenomena. The conduction without decrement. right? So that is what is characteristic of action potential. That in this electrotonic potentials, because they are plateaued, the signal dissipates over this plateau and then it comes back to normal. Right, but over here in action potential, due to these sharp peaks and spikes, the conduction of electricity or electric potential is without decrement in the signal, right? And it does have a refractory period, right? This we will lay more emphasis on in the upcoming slides. So these properties cannot be explained with passive memory membrane properties, and the puzzle scientists for some time, right? What was the key observations that these people had after looking at the action potential? So Hodgkin and Katz were two scientists. They basically saw that the amplitude of action potential is reduced if the extracellular sodium concentration is reduced, right? So it might show a lesser peak if basically the extracellular sodium concentration was reduced. So they thought that sodium influx may be responsible for action potential, right? So this space, maybe the sodium influx is responsible. Also, they propose that depolarization above a threshold can increase sodium conductance, right? So if we depolarize above a certain threshold, meaning that it is going closer and closer to zero, right? So that will increase the sodium conductance because its equilibrium potential is what? It is near positive only, such that it becomes more dominant than the potassium conductance, and V moves closer to ENA, like voltage that is required for this will move closer to the equilibrium potential of sodium, right? They also found that the falling phase of action potential, that is this one, was caused by later increase in the potassium conductance, right? So increase in sodium conductance basically causes this rise in peak of action potential and decrease, and later when sodium channels are all open, then the potassium channels also start opening, because then equilibrium potential for potassium also has to be maintained, and when those start opening, then this peak starts dropping, right? So this is a response of the potassium potential, right? So, there are some very natural questions, that why do we have this shape of the action potential, which is a very, very peculiar shape, and how exactly do the conductance change during the course of action potential in a quantitative manner, right? So how we are saying that initially it is closer to conductance of sodium when it is being generated, and after it is generated, then conductance of potassium is increasing, it is coming down. How are we saying that is happening, right? So let us look at that. So earlier, Hodgkin and Katz had basically observed this, right? Later, Hodgkin with Huxley proposed that conductances could be a function of voltage. That is, the change in voltage could lead to change in conductance, right? To study this, one would want to measure conductances with respect to variation at with time at different values of voltage, right? But if we change the conductance, then voltage will also get changed, right? So that is a problem that they had. So they can check this, but if they are changing one thing, if they are changing voltage, conductance will be changed. If they are changing conductance, then voltage will be changed. Then how will they test this phenomena, right? So they have to vary just one. They don't want both of them to vary. They want only one of them to vary at a time. So how they tested this was, they blocked whatever channel they wanted to check for, right? So what they did over here was, first let us understand the instrument that they had used, and then we will understand how they have done it, right? So they have used a voltage clamp. What will a voltage clamp mean? That the voltage will be constant, right? Or sorry, the current will be constant, I think. in the ionic current is balanced out by compensatory current. So voltage, nahin, voltage clamp means voltage is constant. So first what I said was the correct one, right? So over here current is changed, but voltage remains constant. And the measurement of compensatory current tells the value of the ionic current, right? So whatever current we are giving in compensation to keep the voltage constant is the value which tells us that this much of ionic current is coming in the first place and this much of it we have to balance out, right? So the set voltage is called the command or holding potential, right? Yeah. So this only I needed to see before I said that, but what I said first was correct. That in voltage clamp, actually voltage is what is controlled, right? And like this is constant, right? And current through the membrane is what is measured. And current clamp, the current is what is controlled and voltage is actually what is measured, right? So the voltage clamp can also be implemented using a single electrode for both recording voltage and passing current. Example with a patch recording, right? So over here again, we come to the same diagram where we did say that if this is a plasma membrane, this is the extracellular side of the plasma membrane and this is the cytoplasmic side of the plasma membrane. And if a current starts flowing, it will probably go and first flow through the capacitor. And once some kind of a voltage starts building over here, it will go and it will also flow through the resistor, right? And it will equalize after a while and then the current will stop flowing altogether, right? So what happens is there again in the capacitive current, the voltage is being built across the capacitor, right? But in ionic current, there are different subcategories and that can be looked at, right? So there is voltage gated sodium channel, voltage gated potassium channels, and there are also leakage currents, which means that this is not gated channels. It will be those leakage channels that will allow most of these substances to pass through them, right? So they are passive potassium channels, passive sodium channels, and passive chloride channels, and more like leakage channels, right? So they are free and open, and they will let these things pass through. So in the voltage clamp, the capacitive current is zero, right? So because voltage we are taking as zero, so capacitive current is zero, except when the holding or command voltage is changed, right? Then the leakage current is constant for a given command voltage. So it will be constant only. This is an example of a voltage clamp recording. If there is a small depolarization again, what does it mean? Moving closer to zero, right? Because the equilibrium potential is in negative. So when we move closer to zero, it is going to be depolarized, right? So what will happen if the current change, if this much of depolarization occurs, then, the current changes this much. And I am over here indicates the value of compensatory current, this one, that was injected into the cell to basically revert it to normal, right? But once we actually consider the voltage gated conductances, then the picture becomes more and more complicated. Why? Because it is not easy to figure whether the observed current is through voltage gated sodium channels or through voltage gated potassium channels. And this is the place where some chemicals become very useful, right? So how would they have decided, kind of devised these experiments? It is by blocking some of the channels so that the other channel can actually be studied, right? So that is what they have done further. So for instance, first they have used this particular chemical, which is tetrodotoxin, right? This tetrodotoxin, it has the word toxin in it, right? So it is a poison that is found in the puffer fish. So this is a puffer fish. You might know it from cartoons, if not through zoology and through, the knowledge of fauna in the sea. This is the fish which, when it senses fear, or senses some predator, it basically swells up, right? This, when it swells up, it is difficult for the predator to consume this also, and it will also have this tetrodotoxin that it can release that can numb the predator also, right? So in pufferfish and some other animals, there is a poison which is known as tetrodotoxin that is found, right? So this tetrodotoxin, what it does, it selectively blocks the sodium gated channels, right? So imagine if you were injected with something that blocks sodium gated channels, then you will stop functioning. It will be neurotoxic for you because no impulses and nothing will travel, right, from your one neuron to the other, right? So it will be highly neurotoxic. Sorry. So it will be highly neurotoxic. And this tetrodotoxin is 1,000 times more poisonous than potassium cyanide, right? Again, because of the same thing, because all of your, this thing, like crosstalk between your neurons will be stopped. So your heart beating, your respiration going on, you being able to, like consciousness, memory, speaking, these are all secondary. Even your vital organs might not get the signal, right? So they will also have difficulty in working. So this was actually the chemical that these scientists they used in a synthetic setup, right, in vitro setup. And what they did was they know that it selectively blocks the voltage gated sodium channel. So what they did was uh, okay, what they had done was that they had basically taken that toxin and they had given it to the cells that they were observing. And now they know that whatever response they are seeing is not because of the sodium channels, because they are already blocked. It will be only due to the potassium channels. And then in a couple of slides, we will see what that response is. Right over here, there are also many natural poisons that function by blocking some aspect of the neural activity. Therefore, they can lead to either muscle dysfunction or cardiac arrest or respiratory problems also. Right? Over here, when they did use this tetrodotoxin, what happened was this was normal, right? So normally an action potential where it is attained, sorry, when a good enough depolarization is given, and this can cross a threshold, then an action potential can also be received. Yeah, it can be obtained. So there is a large capacitive current over here. Initially, there will be an inward current, and then later there will be an outward current also, right? As the leakage conductance is constant, leakage current can easily be computed and it can be subtracted, right? Then we can separate the voltage gated sodium and potassium currents if we can block specific channels, right? And this is what will be obtained by using these chemicals. So the voltage gated sodium channel block can be blocked by using tetrodotoxin. Similarly, this voltage gated potassium channel can be blocked by using 4-aminopyridine or tetraethyl ammonium, right? So one of these can block these potassium channels also, right? So what will happen? If, for instance, this is a normal scenario after this much of depolarizing current has been given, right? If there are both sodium and potassium channels, right, then what will be the individual graphs that look like when we measure in presence of tetrodotoxin? When the sodium channels are not functioning, then we will see this kind of a peak for the potassium. And when we measure in terms of in the presence of T, which is something that is going to go and block the potassium channels, then this is the kind of peak that we get for the sodium channels, right? So what we see is that sodium is responsible for bringing it back to normal, while potassium is actually over here. Sorry. So, yeah. While potassium related current is taking it forward, right? So over here, what you can see is that Okay, this we have already studied, right? That the resting voltage in all cases we have taken at around 60, and what is the driving current and the net driving current for potassium and for sodium, right? And you know that at closer to the resting voltage, the chemical driving force for potassium is outward, and the electrical driving force for potassium is inward, right? And the net driving force is also outward, right? As we go to more and more positive, the chemical driving force is always outward, right? Because potassium is much less in the outside than the cytosolic side of the cell. But the electrical driving force, it changes as and when we reach positive potential, right? So it also starts increasing. Then uh, just a moment. Yeah. So then the net driving force will also be increased, right? In case of sodium, what happens is that initially the chemical driving and electrical driving force, both of them are inside when we are closer to the resting membrane potential. As and when we go farther away from the resting membrane potential, these currents, these forces, they start to oppose one another. So the farther away we are from potential, the driving force is majorly potassium, and the closer we are to the resting voltage, the driving force is this thing, potassium, sorry, sodium, right? So that is the outcome from the slide, right? So if we are closer to resting membrane potential, then what is driving the what is the net driving force? It is sodium. And if we are farther away from the resting membrane potential, then what is the major net driving force? It is potassium, right? Then this is how the conductances can be measured from these individual voltages, right? And yeah, the conductance of both sodium and potassium are functions of voltage. Hodgkin and Huxley determine the exact dependence of this. If you depolarize a neuron and don't clamp the voltage, then V will change depending on the GNA and conductance of sodium and conductance of potassium. And for these, set of experiments, they have also received the Nobel Prize in 1963, right? So a good set of studies they had actually done. This is the derivation for that, if you wish to look at it, right? And this is what we just saw, that action potential, it is basically a sum of why we are only looking at sodium and potassium right now, because these are the two most abundant ions that are present, right, inside and outside. So it is a sum of the sodium conductance and the potassium conductance channels, right? The action potential. So that is why that kind of amplitude also it has. So according to the Hodgkin-Huxley model, an action potential involves the following sequence of events, right? A depolarization of membrane causes the sodium channels to open rapidly, right? So what will happen? If it is going farther away from its equilibrium potential, then the sodium channels are the first ones that are going to get open, and they will open rapidly. This will increase, this current causes further depolarization, because sodium channels are opening. Equilibrium potential for sodium is positive 55, around positive 55, right? So they will try to put it to closer to equilibrium potential of sodium. So further depolarization will occur, right? Thereby it will open more sodium channels in response to that, and more of inward current. So this loop will start, wherein more depolarization causing more sodium ions to open, causing more depolarization, and vice versa. This loop is going on now, right? And this depolarization after a while will hit the limit. It will limit the duration of action potential in two ways, right? It will gradually, whatever sodium ions are open, they cannot always remain open, right? They have to close at some time, right? So they will gradually get inactivated, right? Also, when this kind of a change is happening, then the equilibrium for potassium has also now drastically changed, right? So now it wants that it should come back to the closer to the equilibrium potential of sodium of potassium channels, right? And that is repolarization. It is closer to the resting membrane potential, right? So as a result, the inward sodium current will reduce and outward potassium current will increase, which tends to hyperpolarize the membrane. Right. So, for a brief moment, it will go beyond the resting membrane potential also. It will go further negative than that. That is why it is being termed as hyperpolarization of this membrane. Right. So, this is what it looks like. The membrane potential goes below the point from where it had started. This is where it is starting. Right. This would be the straight line, but it is going below this also. Right. It is going around here, that EK, right, closer to EK. So this is known as after hyperpolarization, right? Because it is happening after the action potential. Just a moment. Yeah. So if the membrane potential is determined by the balance between different ions, then the sodium ions are the ones that are trying to take the membrane potential towards their equilibrium potential, while potassium ions are trying to take the membrane potential towards their equilibrium potential, right? Equilibrium potential for in terms of squid giant taxon, for sodium it is near plus 55 and for potassium it is near minus 75, right? So that is, these are the directions they are trying to take it in. So during an action potential, the sodium conductance has increased, but then it also gets deactivated. It will increase sharply, but then sodium channels will start becoming deactivated, right? While potassium conductance increased a little later, it started increasing a little later, but it did not activate, right? So the potassium it did not deactivate. So these potassium channels are active more longer also, for longer. And the longer the time the potassium channels are active, more and more influx of potassium will occur, right? So therefore potassium ions keep going out and the membrane keeps becoming more and more negative, right? Because we have additional potassium conductance here, the membrane potential will go towards the equilibrium potential of sodium, which is minus 75, but not below that. Right? It will only touch the negative 75. It will not go beyond that, because what is the thing that is causing it to go till minus 75? It is a potassium ion itself. Right? As voltage-gated potassium channels gradually close, the membrane potential can return to the baseline also. So this is what happens, right? This is near minus 70, this is minus 55, which is the threshold over here for this kind of neuron. And for the action potential to come in this kind of neuron, right, that also might differ from cell to cell and neuron to neuron. Now, what is the threshold, right? So if the stimulus strength is, sub-threshold, means it will not cross the threshold. This kind of a, electrotonic currents, potentials only we will see, right? If it reaches the threshold, then it will give an action potential. And even if it reaches much more than, almost double than that was required in the threshold, it will not give double the peak. It will still give the same peak, right? So it is just an all and none phenomena. Either it will give peak or it will not give peak, right? Action potential will not have a smaller or shorter peak, right? It will have, it will be 0 or 1. Over here, it is not there, and over here, it is there. That is the only difference, right? So two opposing factors begin to act over here. The voltage-gated sodium channels open, which try to further increase the voltage, and leakage channel try to decrease the voltage, right? Action potential size depends on voltage-gated channels, and it does not depend on the stimulus size, right? It will only depend on how these voltage-gated channels are acting, right? So when sodium channels are inactivated after some time, after an action potential, this is what happens. So this is when they are open, right? And this is when they are inactivated. Once they are inactivated, what happens is it is physically blocked by an inactivation gate, which will no longer let any of the sodium ions that are present outside to cross into the cytosolic side of the neuron, right? Now, this is when this refractory period that we talked about will, occur. Right. So, what is this refractory period? If you remember, it is a state wherein the channel is not active and it cannot be activated also. Right. So, why is that? Because, this is a conductance of sodium. Right. Uh, you can see that if you are giving the same threshold in this much time, right? Say that this time was like 1 second, right? For instance, this time was 1 second. So if 1 second apart, you are giving the same amount of threshold, it will not be able to reach the peak. If after 2 seconds you are giving the same amount of threshold, it will again not be able to reach the same kind of potential as earlier. But if after 3 seconds you are giving, then it will be able to attain the same kind of peak as the previous potential, right? Because the stimulus over here was same in all. But it in case 1 and 2, it was in refractory period. That means that the stimulus that we are applying is the same, but the sodium channels that are available are much lesser, because most of them are in the inactive state right now. They need a little time for them to come back to their normal resting state when they can be activated. And in the third situation, that is the case that is happening, right? Then comes current clamp, right? So right now we were talking about the voltage clamp. Now, with respect to current, what happens? Again, refractory period is the amount of time after a spike in which it is possible or impossible or difficult to get another spike, right? That is the refractory period. This is the refractory period. Out of this, there will be some period that is the absolute refractory period, and some other which will be the relative refractory period. What does that mean? So first, what is the reason for a refractory period? It is basically inactivation of sodium channels or slow closing of the potassium channels also, right? Now, this is how propagation of action potential occurs along a axon, right? So, over here again, we can recall the time constant and the length constant, right? Axon hillock is actually the place wherein an action potential is generated. Why is this the place where action potential is generated? If somebody has studied that or has thought about it, why not over here? Why not in the cell body itself? Why not in the dendrite itself? Why not directly in the synapses? Why is action potential generated in the axon hillock? So this actually happens because the maximum number of channels are over here in the axon hillock region, and over here the threshold is minimum, right? So the threshold to reach this action potential is also less. So this hillock has high density of voltage gated sodium channels, which is the most important reason why axon hillock is where the nerve impulse is getting generated, or these action potentials are getting generated, right? So if this is the site wherein it was generated, that is site A, right? Then this will have the region of depolarization, and the region next to it on both sides will be normal, right? But what will happen is that at the resting phase, the direction of there will be a direction that will be chosen initially, right? Uh, yeah. So this will be the direction of action potential propagation. Now, in this case, why it is moving forward? Because over here again, the channel density might be a little, closer to the channel density in the axon hillock, while over here, maybe the channel density is very, very sparse, right? So that is why it will, proceed in one direction, and similarly, it will keep on going in that direction. Now, why it will not from site B, why will it not come back to site A when we clearly know that site A has maximum density of the voltage gated sodium channels, then why it is going to C and why it is not coming to B. Now that is because this site A will have sodium channels inactivated at the time when B is transmitting it to C, right? So not all of them have recovered and they are actually in the refractory period. So the overall, available number of channels that will be there will be more in C only. That is why this is very, very, unidirectional. That is why the direction over here doesn't change. It is unidirectional because it cannot go back, because over there refractory period is still going on and it cannot turn back, right? Yeah. So then we can also talk about saltatory conduction, right? Saltatory conduction generally occurs in case of myelin, right? So why does it occur in case of myelin? What is myelin? Myelin is basically a sheath-like, of white substance that is secreted by oligodendrocytes, and it is a sheet that is insulating the axon of the neuron, right? So why is this myelin important? Because like we have insulated wires that can carry the electricity very nicely, right, without leakage of electricity into the outside world. Similarly, this myelination acts as insulation on the neuron, axon of the neuron, through which it can carry the currents, carry the voltage or the impulse more smoothly. Right. So what happens is these are not present as a continuous, sheath. Instead, what happens is it is present as, they have breaks in between, which are known as nodes of Ranvier. Right. So what happens is that wherever this myelin is present, the resistance on the surface is more, right? So axial resistance over here is more, due to which current will not leak from the surface, but it will keep propagating into the forward direction. Right, but then this current will also dissipate because there is a length constant also that is involved. Right, there is only a certain length that a current can travel without getting regenerated or rebooted, right? So that is why these nodes of Ranviers are there. These are again places wherein there are these are hotspots of channels, right? So once the potential reaches over here, again there will be depolarization of channels, and due to which action potentials will get reboosted, and they will be present again, and then it will move further, etc. Right? So that is what will happen. And you can see that in this, graph, which plots the distance along an axon in relation to the time between stimulus and arrival of action potential, the place where this myelin, the node of Ranvier is present, is actually where it is taking a jump. Then it is just getting conducted. Then at the other node of Ranvier, again, this is taking a jump. Then again, it will just be continued, and so on, right? So variations in the properties of voltage-gated channels can increase the signaling capabilities of neurons themselves, right? So there is a rich variety of voltage-gated ion channels that are present. The gating of voltage-sensitive ion channels can be influenced by various cytoplasmic factors, and the excitability properties vary between regions of neurons, right? So as we said, that axon hillock is a densely populated, sodium gated channel region. That is why there will be more excitability over there than, say, the, the cell body or the dendrite. Then the excitability property also varies among neurons. Different kinds of neurons will also have different kinds of densities of these various channels, right? So further, we'll continue in the week 4 one that I have in a separate presentation, right? Just give me a brief moment. Yeah. Okay. I have around 140 slides. I'll try my best to finish it, which is why I'm going fast also. Let's see how much we can cover. We'll be able to cover it. No worries. Yeah. Yeah, so this part we have already studied, right? Yeah. So what is the relevance of these action potentials, right? So how are they relevant? They are relevant because when these action potentials are actually reaching the synapse, that is the end of one neuron and the beginning of another neuron, right? So the junction between these two neurons, when it reaches that point, results in release of neurotransmitter. So that is the result of or relevance of action potential, that it is transmitting a nerve impulse, which when it reaches these synaptic end buds, they result in release of neurotransmitter, which carries information to the the other neurons in the vicinity, right, or the neuron with which the synapse or synaptic junction is being created, right? So generally, the graded potential do not result in release of neurotransmitter, but there are some exceptions sometimes that are seen, right? Then there was this very famous, neuroscientist who is Charles Scott Sherrington, right? And he received Nobel Prize in 1932, and he was the one who coined this term synapse, right? And synapse is one of the most integral parts of how neurons are functioning, and how, a few million neurons have, billions of different kinds of connections and things that they remember and do, right? And there is basically this organization named as Neurotree, wherein multiple, starting from Sherrington, then his disciples, then disciples of their disciples, and like that, everybody is arranged. And it is actually interesting to know that Nitin sir is also a part of this neuro tree. So if you, search for Nitin Gupta, it will give you his institute. He's at IIT Kanpur. Then further, because he has also now trained students, and has disciples of himself, right? So those students are also a part of this neuro tree now, the people who are working in his lab. Right, so they are all somehow the descendants of, like science wise, they are descendants of Sherrington, right? So Nitin sir's PI, for instance, in during postdoc was, trained by somebody else who had some relation to some of the disciples of Sherrington, like that it is going on. Right. Similarly, there is actually another professor in our department, Professor Arjun Ramakrishnan. He also has his mention over here because it is a tight knit community, right? So these people who are working on neuroscience, neural networks, cognition, and this aspect, they are basically synapses. They are part of this neurotree somehow, right? It is very interesting. Yeah. So this basically, tells about the various different kinds of nerves, and how these nerves are going to various organs, and how various organs are basically being, controlled by the brain, right? So there was another scientist, Otto Loewy, who showed that a chemical which was later found to be acetylcholine, which is a neurotransmitter, conveys signal from the vagus nerve to the heart, right? So this vagus nerve, which is coming out of this medulla oblongata region of the brain, it is basically, it is going so many places, right? It is stimulating the heart, it is going to the lungs, to other alimentary canal organs, etc. Right? And through how it is doing this? Through basically a neurotransmitter, which was acetylcholine. So how he basically described that was, he he wanted to see that sometimes, like how this observation began was, that when a person dies, the heart sometimes, obviously the death has to be of some other cause, not something of a heart related issue, right? But if you, for some moments, if you open the, autopsy the person right there, sometimes the heart seems to be beating, right? That doesn't mean that the person might be alive, but it sometimes happens because of, the a leftover, neurotransmitter that is still flowing, right? So in case of humans, I'm not sure how common this phenomena is, but it happens very frequently in case of, mammalian models, right? So for instance, I work with mice, right? In case of mice, sometimes it happens. Like earlier, when I was not aware of this phenomena, I also used to wonder, why is this happening? Did I not, so there are different ways of euthanization, and one of them is cervical dislocation, in which you are breaking the connection of the spine to the brain, right? In that case, you cannot survive, right? So, in case that was also done, and when you opened up the this model organism to actually then isolate organs and to perform whatever experiment you had to, sometimes it used to be like most of the times actually it used to be very intriguing that there is this dead mouse but the heart is still beating, right? So how does that happen? So these kinds of observations actually intrigued Otto Loewi as well, right? So what he did one day, he basically stimulated the vagus nerve from the donor's heart, and there was some kind of a heart rate slowing that occurred, right? And once this occurred, what he did was the fluid around this heart that was there, he removed that, right? And there was that recipient heart which was normal, and he added this fluid to that heart, and it also beated slowly, right? The heart rate slowing occurred over here also. So he basically, came out with this theory that it was the fluid around the heart due to which the chemical around the heart is causing it to decrease its heartbeat, right? In 1936, along with Henry Hallidale, Otto Loewi actually got this Nobel Prize, right, for the discoveries relating to chemical transmission of nerve impulses, right? So over here, despite like apart from electrical impulses, there was also chemical impulses that was involved, right? Because there is a chemical that is being released, and that over here is the neurotransmitter, right? Then Lewis' discovery sparked a lot of debate in 1930s over how chemical signals could generate electrical activities at other synapses. Right, so there were two schools of thoughts that came out of this debate. One was a physiological and one was a pharmacological. Right, in case of in 50s and 60s, there were improvements in technologies which actually showed that both types of synapses actually existed. There were chemical synapses also wherein neurotransmitters were involved, and there was electrical synapses also wherein there was a small connection between this neuron to that neuron, and it would transmit the nerve impulse directly, right? So there were current pathways at electrical synapses, that is a connection of sort, and current pathways at chemical synapses was through neurotransmitters, right? So the type of synapse and the different properties are mentioned over here. The distance between pre and post synaptic cell membranes in case of electrical synapses is very less. It is 3.5 nanometer, while in case of chemical synapses, it can be much higher, around 20 to 40 nanometer. In case of cytoplasm, there is a continuous cytoplasm over here in case of electrical synapses, whereas it is non-continuous in case of chemical synapses. Then, if we look at the ultra-structural components, that is, if we go at the structural details, then you can see that over here there are gap junctions that are present in electrical synapses, wherein there are presynaptic vesicles and active zones over here, and postsynaptic receptors that are actually present in this chemical synapse, right? What is the agent of transmission? In electrical, it is ion current. In chemical, it is chemical transmitter, right? Then the delay is virtually absent in case of electric synapses, and it is again very less, around 1 to 5 millisecond in case of chemical synapses, right? And the direction of transmission is usually bidirectional in case of electrical, but it is always unidirectional because there will be one synaptic end bulb and one dendritic end with all these neurotransmitter receptors that is present, so it will always be unidirectional. Yeah. So, when we look at crayfish and we see that electrical synaptic transmission was first described in the giant motor synapse of this crayfish, right? So, before this, only the chemical synaptic transmission was known of, right, by that Otto Lewis experiment, right? And different versions of that experiment only later. So, in this, there was a kind of electrical setup wherein there was a current injection in the pre and postsynaptic neurons, and then there was an electrode which used to measure this, right? So in case the current was injected in this post one, there was some changes in measurement in presynaptic electrode also, and in case the injection was in the presynaptic axon, then also there was some kind of a change in the postsynaptic electrode potential also. So that is how we know it is bidirectional also, right? So the electrical synaptic transmission is happens somewhat like this. This is presynaptic and this is postsynaptic, right? So electrical transmission is nearly instantaneous, right? So even sub-threshold amounts of current in presynaptic cell can depolarize a postsynaptic cell through electric synapse, right? So in electrical synapse, actually, action potential is not entirely important, right? In case of this thing, post Okay, one second. What am I saying? Yeah, it is not entirely important, right? Even if there are sub-threshold potentials, thresholds that cannot reach the value of this thing. Uh, the threshold value so as to obtain an action potential, it will still be able to go through an electrical synapse. However, it will not be able to go through an chemical synapse, because the action potential is required for the synaptic end, the synaptic bulbs to have those, neurotransmitter vesicles fuse into the end bulb and then get released, right? So that phenomena is not required over here. So that is why even a sub-threshold level can still, get transmitted. So electrical synapses will transmit both depolarization as well as hypopolarizing current, but they will have some reactivation, but some will have some rectification, right? So this, for instance, is a plasma membrane, right? This is a structure of electrical synapse. And specifically the gap junctions of these electrical synapses, right? So you can see this is the presynaptic cytoplasm, this is a postsynaptic cytoplasm, right? So this is the up, like top view. This is not the synapses per se. This is just the receptors on this side, right? Now there could be another view wherein we section. Yeah, this is actually showing the channel or the gap junctions. The gap junctions due to which the channel is formed by the pores in each membrane, right? So these pores have a diameter of around 1.5 nanometer. Regarding this also, we did one question last week, right? So these individual proteins that form up this channel in one segment or like one neuronal membrane is known as a connexin. These connexins, 6 of them come together and they form a connexon, right? And they basically have extracellular loops for homophilic interactions and transmembrane proteins also, and presynaptic side. That is it, right? And they have cytoplasmic loops for regulation also, right? Now, what will happen is, yeah. Uh, what the question last time was that in this electrical synapse, how many of these connexins are actually present, right? So there are 6 and 6, 12 of these connexins present. How many of these connexons are present? 2 of these connexons are present, right? So those kinds of questions can be asked. Then electrical transmission allows rapid and synchronous firing of interconnected cells, right? An example is the sea organism Aplysia, right? Now, what it does is that it has an ink gland, right? So as soon as it gets a sensory stimulus that, it has to defend itself from something, all of a sudden, this electrical transmission of the signal occurs, due to which inkland instantaneously almost releases ink, so that the predator, he will be lost, and he won't, or confused, right? Yeah. Then we come to chemical synapses, right? So there are again some parts in this chemical synapse. One is the synaptic cleft, which is this region in the middle. It is the region separating the presynaptic and postsynaptic cells at a chemical synapse, right? Then there are neurotransmitters. These are a chemical substance which are released from presynaptic terminals that will bind to specific receptors on the postsynaptic cell membrane. Then there are synaptic vesicles also. These are presynaptic terminal contains the presynaptic terminal contains discrete collection of these vesicles, right? You can see over here there are larger vesicles in which these neurotransmitters are stored. And these are the synaptic vesicles basically, and neurotransmitters are released when these vesicles get fused to the plasma membrane. And there are active zones, the regions of the membrane specialized for release of neurotransmitters. So this is an active zone, this is an active zone, right? And there are also then neurotransmitter receptors which are present on the postsynaptic neuron, right? Yeah. So over here, you can see a detailed image of synaptic vesicles in through electrical electron microscopy, right? So this is an axon. This is the end bulb of the axon. These are synaptic vesicles, right? This is the other, neuron, the dendrite of the other neuron, and over here there will be some receptors. This is the synapse in the middle. This, gap. This is the gap wherein these synaptic vesicles will release the neurotransmitter, and this neurotransmitter will be uptaken by this the neurotransmitter receptors that are present on this postsynaptic neuron, right? So this is the dendrite of the postsynaptic neuron. The dark arrows indicate active zones. So this is where these are fusing and they are getting released. So that is why it is the active zone. Then the action potential causes voltage calcium ions to open and allow influx of calcium. Right. So what happens is when action potential is transmitted and it comes to the end of this, synaptic end bulb, what happens? It opens calcium gated channels, and there is a flow of calcium that occurs over here. Right. Due to which what happens? The abundance of calcium inside causes synaptic vesicles to fuse with the membrane and release a neurotransmitter, right? Then neurotransmitter then diffuses into the postsynaptic membrane where it binds specific receptors and results in opening of the channels, thus changing its membrane potential, right? Then this neurotransmitter, it is not just released into the synaptic cleft. The synaptic cleft has to be left vacant also in between, right? It cannot be constitutively active. So that is why there is removal of neurotransmitter from the synaptic cleft as well. Right. So what happens when this has to occur? There are multiple ways through which this neurotransmitter can be removed. It can be degraded, it can be recycled, it can be reuptaken. It can be absorbed entirely by these dendritic, receptors, etc. Right. So one phenomena is reuptake, wherein there are transporter proteins on the presynaptic membranes itself. Right. So there are reuptake proteins that are present itself, which can reabsorb the neurotransmitter, which is then recycled into the synaptic vesicles, and next time they can be released. Right. Then there is enzymatic degradation, right? That, for instance, if acetylcholine is a neurotransmitter over here, then it can quickly get degraded by an enzyme which is known as cholinesterase, right? So that can also be used. And then comes diffusion. So they can move out of the synaptic cleft and eventually get absorbed somewhere else, or they can get secreted out, right? Then we come at the postsynaptic potential, which is the PSP. Please give me a moment. Yes. Okay. So then we can talk about the postsynaptic potential as well, right? So there is a presynaptic action potential that happens, right? And after this, this has gotten transferred along to the other neuron, to the postsynaptic neuron, then what happens? The activity that is caused in this postsynaptic neuron is actually called the postsynaptic potential, right? Now, what happens? It could be excitatory or it could be inhibitory also. So, if it is excitatory, it is excitatory postsynaptic potential, EPSP. If it is inhibitory, it is inhibitory postsynaptic potential or IPSP. Right? Whether it is excitatory or inhibitory, how will we know that? It will depend on which channels are opened by the neurotransmitter. Right? So, for instance, it is opening more of, sodium channels. It will probably be excitatory. If it is opening more of, say, potassium channels, it will probably be more inhibitory, right? Or chloride channels, for instance, right? So this is the excitatory postsynaptic potential, right? So it is still going towards depolarization over here, right? So the synaptic delay can be used. This is the delay, right? It can be used to tell whether two neurons are connected to each other or not. So in case of electrical synapse, for instance, this delay will almost be non-existent, right? But in case of chemical synapse, this delay will be there in milliseconds only, but it will be there, right? So we know that they are not connected to each other, right? Or how far away they are from each other approximately, right? So why is chemical synapse even used if there is an option of electrical synapse, and why at all do we use chemical synapses? Because over here, how much signal has to be given, that is tunable. So the amplification is tunable, there is flexibility, and there is filtering also, right? Only the more important signals get through, not all the signals get through, right? Otherwise, the brain will always be in an overactive state, right? Then let us go through some common neurotransmitters, one of which is glutamate. It is the most common excitatory neurotransmitter, right? It is what? Glutamate, right? Then comes GABA. That is gamma aminobutyric acid. It is the most common inhibitory neurotransmitter actually that is present. Right. Then it is acetylcholine, which is very common in the motor neurons. Right. Yeah. So if we look at these three itself, those three, they were neurotransmitters. Then there is another term which is known as neuromodulators, right? These are mainly dopamine, serotonin, noradrenaline. They cause large scale modulation of brain activity, not just, one neuron to other neuron, but large scale modulation. That is why they are known as neuromodulators instead of neurotransmitters themselves, right? So dopamine, serotonin, you might have heard about these, right? The happy hormone, the anxiety hormone, etc. Right? Then the neurotransmitter can open channels on postsynaptic neurons directly or indirectly, right? So how does this happen? If it is an inotropic receptor, which is ligand gated, for instance, maybe the neurotransmitter itself is the ligand due to which it can open the ion channel, and there is flow of ions that happens across the membrane over here, right? Sorry. Yeah. If it is metabotropic, such as G protein coupled receptors, maybe the neurotransmitter binds to its receptor, due to which there is some change over here. Maybe G alpha gets separated or G beta gamma gets separated, and this goes to a nearby receptor, and it regulates this receptor's opening, due to which again ions can flow inside, right? So when we talk about these different kinds of neurotransmitters, they can have different kinds of receptors also, right? For instance, glutamate, which we said was the most common neurotransmitter, it has inotropic receptors as well as metabotropic receptors. The metabotropic receptors are GPCRs themselves, but the inotropic receptors are varied of variable kinds. Yeah, the NMDA receptors, the AMPA receptors, and the kinase receptors. All of these are cation channels which are permeable to potassium and sodium, and the equilibrium potential is near 0 millivolt, right, for these receptors. Then comes GABA, right? It has an inotropic receptor, that is GABA receptor, which has basically equilibrium potential closer to -70, right? And the other, which has a GABA B receptor, which is metabotropic receptor, which activates the potassium channels. And this receptor would cause an IPSP if the voltage was more than -70 and excitatory potential if it was less than -70, right? So depending on excitatory or inhibitory will also depend on how much is the resting potential itself, right? Then we talk about synaptic integration, right? Synaptic integration, plasticity, etc. are multiple terms that are used in case of pathology also. So what is a synaptic integration? When a neuron receives inputs from multiple other neurons and its activity depends on the net input received, right? The presynaptic neurons may give excitatory or inhibitory inputs to a postsynaptic neuron, and a single postsynaptic neuron may receive both inhibitory as well as excitatory inputs, right? So that is true, right? One neuron is not doing one kind of a function. It is not just giving out one kind of a output, and it is also not receiving only one kind of an input, right? That is what it says. Then this is a knee-jerk reflex. I am sure you might be already aware about this, and we have also discussed it in one of these, question answer series. We can discuss it in terms of IPSP and EPSP over here, right? So what happens over here is that but whenever we get a signal, the quadriceps take the signal through the sensory neuron to the spinal cord. There are motor neurons in the spinal cord that are present, and these motor neurons will take the signal back to like they will read the signal, they will take the signal back to the quadriceps so that it will like stretch. On the other hand, what will happen? Like it will contract actually so that this will move forward. But for this knee to move, like for the leg to move upward, not only this muscle, but this bicep or flexor also has to work, right? So simultaneously, when the signal is being carried to the quadriceps, there will be an inhibitory signal also that will be given to the flexor motor neuron, which will go to this bicep or flexor muscle, which will cause it to relax. So this muscle is contracting, this is relaxing because of the same signal, and two different kinds of postsynaptic potentials. This is giving a excitatory postsynaptic potential, and this is giving an inhibitory postsynaptic potential, right? So that is what is being said over here. Then if an inhibitory input is given, then the firing of neuron can also be reduced, right? So without inhibitory input, it might go like this, but with inhibitory input, whenever it is given, obviously it will negate the excitatory input or the threshold or like the potential that is being received. That is why these kinds of scattered peaks are then seen, right? Then this is another way of explaining and synaptic potential and resting membrane potential, the kind of threshold and action potential, right? So what is important for you to understand over here is that the peak synaptic potential you can see is higher over here in the dendrites, right? Over here, the synaptic potential is actually higher, while the synaptic potential is actually gradually decreasing as we are increasing our distance from the synapse, right? It makes sense because synapse is the one that is receiving the signal. So the highest potential is over there only. As it moves from the synapse across the cell body to this axon, it is already getting decreased, right? But why are we not seeing action potential over here in the synapse itself, and why we are seeing it in this axonal hillock region? Why is that happening is because the threshold that is there is also going down, right? Over here, the threshold for the action potential to be, put up is very high, but over here, the threshold actually is going down along with the synaptic potential itself. So, an action potential can actually be reached in this region of the axon hillock, right? So, that is also another point. So, why this threshold will be higher over here? Because probably the number of sodium gated channels are lesser over here, and number of sodium gated channels are much more over here, right? So, that could be one reason for this threshold being up. Then there is another phenomena which is known as temporal summation, right? What is this temporal summation? When two synaptic inputs at different times can produce a summation effect if the delay between the two is small relative to membrane time constant of the neuron, right? So there are two synaptic currents that we give, but what happens is that the duration is such that in when this current was almost wearing off, this other current is given. So, like individually, they would have gone up this big only, but because they got a little help from the previous current, only this much had gone down, and it had to rise from here only and not from here. So it rose above the threshold, and it gave an action potential there, right? So that is basically temporal summation. Then there could be spatial summation also, right? So this is with respect to space when it happens. Like when there is a long length constant, then it might happen. In case of a shorter length constant, it is difficult to happen, right? So then there could be other kinds of synapses also, such as spine synapse. Instead of to the dendrite, they are giving it to the spine or to the shaft region of the cell body, right? So shaft synapse and axodendritic synapses can also be there. That instead of synaptic end bulb on the axon, it is directly on the axon that the dendrite is present, right? So yeah. So this is axodendritic synapses. Then there could be axosomatic synapses wherein they are near the soma, that is the cell body, and axoaxonic synapses wherein one axon is in close vicinity to the other axon of the neuron and they are forming a synapse between one another, right? So the synaptic contact can occur on the cell body, the dendrites or the axon of the postsynaptic cell, right? It can be anywhere. Then we begin with sensory systems, right? So do we have time to cover it? Let me just see. So anyway, I told you we'll have one more class. So we have 70 more slides to go through. I really doubt my capabilities of it being able to be finished by 9 at least, right? So okay, we'll first go through the question answers itself. Although we might not have touched a lot of those concepts till now also, but first let us just finish that at least so that your question answer part is done and you are prepared with all your PYQs at least. And then that portion along with the revision, maybe we can take up in the extra session that we are going to conduct, right? So, yeah. So the part that is actually left for discussion is the sensory organs, the different kinds of senses, the ones that have been, dealt with in detail are the auditory ones, the olfactory ones, the visual ones also, right? Gustatory, I don't remember, but maybe I can check, right? So for now, we can go through the previous year questions. There'll be around 10 questions and we can go through the questions and the answers, the rationale behind them if we have not already studied it, right? So first question is, a single action potential usually releases neurotransmitters from one synaptic vesicle, right? Is the statement true or false, right? So you have to remember that the synaptic vesicle is, the action potential, the chemical synapses are actually used because it can be uh, tunable. The amplification can be tunable, right? So amplification will be able to be tunable if we have control over how much synaptic or how many synaptic vesicles are actually getting fused. So this statement is actually false, right? So action potential can cause release of neurotransmitters from thousands of synaptic vesicles also, from hundreds also, from just five also. It depends, right? Then in a chemical synapses, communication happens by the movement of charged neurotransmitters from one neuron to the cytoplasm of the second neuron. Now, is that what happens? Is it true or is it false? Do neurotransmitters actually move from one neuron to the other neuron? It is false, right? So, the neurotransmitters are getting released. They are probably acting as ligands for, some kinds of, ions, some kinds of stimulants for opening ion channels, which are then, these ions are actually what is going inside, right? So communication happens via release of neurotransmitters into the synaptic cleft, where they bind to receptors on membrane of second neuron, but they do not enter the second neuron, right? They are just on the membrane, right? Yeah. Then the proprioception is a sense of, we have not come to this, but I hope you'll know this. So proprioception is a sense of posture and movement of body parts, temperature, itch or thirst. Which out of these is true for proprioception? So it is posture and movement of body parts, right? So it is the ability to sense the posture as well as movements. Then which of the following is most commonly thought to be involved in signaling of reward, right? Is it dopamine or serotonin or acetylcholine or glutamate? So the reward one is actually dopamine, right? Dopamine plays reward in as a role in the reward system of the brain, right? Serotonin is a happy hormone. Acetylcholine is the one that is involved with the muscles, and glutamate is the other excitatory and the most common excitatory neurotransmitter. The rods, which are the cells in the eyes, respond to equally respond equally to all wavelengths of light. Is it true or is it false? So it is also false, right? So although they are not as distinguished as cones, there are at least three types of cones that are there for different kinds of wavelengths of light, but uh, they will also not respond equally to all wavelengths of light. Otherwise, we will not be able to distinguish in the dark also between different objects, right? So they have a longer range actually, the rods. They range from somewhere in the ultraviolet to somewhere in the red region, right? So they have a much longer range. They have a tuning curve which peaks around 500 nanometer, right, over here. Then the olfactory stimuli have less spatial information than vision. Is this true or is it false? Now, this is an instinctive question also. It is true, right? So, like visually, you can determine more things that, okay, this is placed over here, this is over here, this is over here. By smell, for most things you will not be able to tell, right? Because there are so many, like especially if so many things are that have a distinctive smell are kept together, you will not be able to probably spot where each smell is coming from, right? So less of information about location of olfactory objects are there as compared to visual objects. So suppose we are doing an electrophysiology experiment to measure the responses of projection neurons to odors. We expect to see the smallest change in the observed temporal spikes for which of these? For different trials, for different odors, or for different neurons? This is a very, very scientific kind of question, right? So what they are saying is electrophysiological experiment, meaning they are inserting an electrode, right? In some projection neuron, to some projection neuron, and they are giving some odor and they are testing what is the response of this particular neuron to this odor. So where will we see the smallest change with time with respect to time of the spikes, right? Is it with different trials? Once I do it, then again I do it, then again I do it, or with different odors. That once I'm giving a citrus odor, once I'm giving a very pungent odor, once I'm giving a very fruity odor, right? Or with different neurons. That once I'm doing it for the mushroom body, once I'm doing it for the Purkinje, once I'm doing it for the projection neuron, etc. Right? So which one will have the least change with respect to temporal patterns? It will actually be different trials, right? Because different odors will obviously elicit different response. Different neurons will have different responses to those odors. But once we do multiple trials, it is going to show the least amount of difference in the pattern. So the temporal patterns depend on the odor and the neuron being measured. The different trials or repeated experiments with the same odor and neuron should cause the least change of all, right? Then which of the following is the least accurate about visual perception? A, that visual perception involves many regions in the brain. B, the visual system ensures that visual perception is identical to the image formed on retina. Visual perception can be affected by prior experience. Or D, artificial stimulation of photoreceptors can trigger visual perception. So which of these is least accurate? We have to find out, right? They may or may not be accurate up to some extent, but which of them is least accurate? So this is the one. So visual system ensures the visual perception is identical to image formed on retina. That is not always true, right? That is the image form, right? Perception is something that is also involving some other, parts of the brain, and they are thinking, or okay, this is like if I see two dots and half a circle below it, right? So I am seeing two dots and half a circle below it, but it will look like a smiley face to me, right? Because that is what my prior experience has been with smiley faces and how they are drawn. And some other part of my brain or some other thing, this visual perception area is linking. It is not just, replicating what is there on the retina, right? It is also involving some kind of cognition and some kind of giving some kind of uh, uh, you know, uh, motive to or meaning to what we are seeing, right? So, for instance, this was a very interesting example. Over here, what are you seeing? There are like some three quarters of a circle, of three circles that are made, and there are three brackets that are given over here, right? Half triangles that are given over here. So, but when we look at it, how it looks like is that there were three circles that were first kept, then there was a triangle, and then that is superposed with another triangle. So that is why it looks like two triangles and three balls, right? So the visual perception basically involves many levels of processing and abstraction by the brain, and therefore the perception can be different from the actual image, as exemplified by various illustrations during Sir's PPT also. Then another question is, pheromones are most likely to be involved in which of the following cases, right? In the attraction of female flies to male flies, in the attraction of female flies to humans, in attraction of female flies to the flowers, or the attraction of female flies to the banana. Which of these most likely have the involvement of pheromones? So pheromones are basically chemical substances that are recognizable between the same species, right? It could be the same sex or the different sex of the same species. So that is why the first one is the correct answer. That is attraction of female flies or male flies to the male flies. So pheromones are involved in communication between members of the same species. That is why, right? Then question number 10 is glomeruli are present in the olfactory bulb. in the olfactory epithelium, in the antenna, or the mushroom body. So which of these regions actually contains the glomeruli, which is a tuft of neurons, basically like postsynaptic ends of some neurons, presynaptic ends of the other neurons, that come together and that are taking the olfactory stimulus to the other higher order neurons, right? So these glomeruli are actually present in the olfactory bulb themselves, right? So this is what? This is the olfactory bulb region. These are the different glomeruli, and this is the one neuron. This is the other neuron which is carrying it to the piriform cortex region of the brain. So olfactory bulb includes various glomeruli which receive sensory information from the sensory neurons. Okay. Yeah. Okay. So that is the end of this part. The previous year questions, right? Let me see whether I'll be able to cover some of this part. Okay, first one is visual only, and that is very big actually. I think it will take me some while to go through it. Okay, I will stop sharing the screen actually then. Yeah. So are there any questions I would like that you would like to ask? Pertaining to this session, there is one thing that like the scheduled sessions that we had were till this Monday itself, right? The meeting link will remain the same, but the timing for the extra session has and the date for it also has still not been decided, because we have actually not officially been asked by NPTEL. So they actually send out a circular wherein we give the time and date. So because they have not asked, and they also provide a window of dates during which we can do it. So I don't know the window also as yet. So I don't know when the extra session is going to be held. But you will definitely receive a notification for that, and we'll finish off this thing, and we'll probably revise the questions again, right? So that is what we'll do. And if there are any queries from your end, because I think it will be closer to the exam, if you guys are actually applying for the exam, it will be closer to that. So if there are any other questions that you have, then we can, discuss about that also. So we can try to sort it out. So if not, then for now, I will end the meeting. Thank you so much for joining. Have a nice day. Have a nice night. Good night. Yeah, I'll just stop the recording. 1 second.